And in 1952, you go to University College London.
Right.
We'd said you were going to move to Medawar's department.
Yes.
Let's
yes.
take that in now. It would be nice to know about the research you conducted, how that came about, what you selected to study.
Okay, well, we applied to the Agricultural Research Council for a project grant, and if I remember rightly, the grant application was entitled Extra Chromosomal Inheritance in Mice. And the reason for that topic was that a fellow student of mine back in zoology days called Tony Milne, who always had an eye for odd things in the literature, had come across this maternal effect in the literature, where two inbred strains of mice, which differed in number of lumbar vertebrae, that's the vertebrae between the ribs and the sacrum. One had 5, the other had 6 lumbar vertebrae. In fact, it's a question of whether the 6th is sacralized or not.
Right.
And in the crosses between these two strains, the progeny significantly resembled the mother, always. And he mentioned this, and this seemed to me an interesting interaction between genetics and development, since I'd been frustrated over my Geyer and Smith and the lens antibody story.
You moved, therefore, to fascinating developmental and environmental issues.
The question was, which particular bit of environment? Because it could be that it was a postnatal effect through lactation, though that isn't very likely because the vertebral pattern is laid down pretty early. Or it could be a uterine effect, something exerted through the uterus of the mother during gestation, or it could be something in the cytoplasm of the egg already laid down during oogenesis, during the development of the egg and present in the cytoplasm. And we knew that the technique of embryo transfer, transferring early embryos from one female mouse to another, had been successfully done in the States some years before, and that was the obvious technique to use to distinguish between the cytoplasmic effect and the uterine effect. The milk effect was easy to dispose of quickly because of cross-fostering, so forget about that. But we did the crosses, and we confirmed that indeed between these two strains, C57 black and C3H, there was this difference in vertebral type, and 2/3 of the progeny in each of the reciprocal crosses resembled the maternal strain. So then we took the F1 hybrid embryos and swapped them over between the females of the strains and looked at the vertebral type of the young. But that sounds easy and straightforward, but it took a long time, because first of all we had to get the embryo transfer technique going. In order to do that, we obviously didn't use inbred strains because they don't breed well. You have small litters, they're not very fertile. We used good random bred strains, and we induced ovulation with hormones so as to get more eggs. And in the course of doing that, we did work on the actual technique, how the number of eggs shed related to the amount of hormone given. That was interesting to the Agricultural Research Council, of course,
so this is a
who
study
were
of
paying us.
gonadotropin, right?
Yep, that's right.
Yeah.
And what would happen if you let those super-ovulated mice go on and get pregnant, super pregnancies, we looked at that. We looked at the relation between fetal weight and placental weight and litter size.
It's like
was a lot
opening
of
an
work.
Aladdin's cave of research opportunities.
That's right.
Can we take in some of those?
Yeah, sure.
Probably. Let's just go back to the actual transplantation, because you say this was a skill really that took time to get.
It took time to get, and it took all these other things to be established first.
So they were all going around the same time?
Yes, that's right. That's right. And it wasn't until right at the end of the 1950s that we finally solved the problem and published the paper on the basis for the lumbar vertebrae maternal effect.
So what
but
we're
throughout the
actually-
1950s, we were publishing papers on super pregnancy and the technique of embryo transfer and that sort of thing.
Yeah. So we're talking about 5 years, 6 years of actually working.
We actually worked together in London for 7 years, but it would have been like 5 years
Getting
before
the
we got an
techniques all
answer,
sorted, and then you
answer
could
to
go
our
on with the
original
main
question.
The main question. Yeah.
That's right. Yes,
Yeah.
yes. And we were fortunate because very often one only gets a 3-year grant. Well, in those days, one only got a 3-year grant from the Agricultural Research Council. It would now be called a postdoc. But in fact, we got a total of 7 years. And at the end of that time, of course, we had to look for proper jobs.
This was because the research was actually producing
Yes.
such a range of
That's
publications
right.
that
Yes.
they felt they had to fund you on.
Yes.
Yeah.
At the end of 3 years, we moved from University College to the Royal Veterinary College because we had so many mice. We'd outgrown the mouse facilities at University College.
This was
Our little
Haldane's
mouse room.
fly room. Was this a...
It was a room in Haldane's fly house, yes.
Yeah, and he said you'd taken over.
We had too many mice.
How
There
did
was-
you negotiate that transfer to Camden? Was that to the Royal Vet School? Was that easily done? Did they invite you? I mean, I just wondered why you...
I guess we looked around for anywhere in London that
there were
would have mouse space.
Right.
And the Royal Veterinary College had surplus mouse space.
Yeah, and you had four good years
And
there.
I think we arranged it through Professor Amoroso, who was the professor of physiology there, but we weren't in his department. Fortunately, Donald realised that it would be very much to our benefit not to be in any particular department. And it turned out that that was very, very wise because the departments were all at loggerheads with one another. And as a separate little
You could
unit,
stay quite clear of that.
we could stay clear of that and we could use the facilities of all the departments. We
Yeah.
used the controlled temperature rooms from the animal husbandry department, the X-ray equipment from another department and so forth.
So you kept out of the crossfire,
Yes.
but managed to
As
take
far
on
as we
all
could.
the advantages and
As
have
far
a
as we
good
could.
relationship with the places.
As far as we
Obviously
could.
were good years, yeah.
Yes.
Yes. Just looking at the domestic scene, because that was an important time for you in your marriage. Whilst we're still at University College London, we should say that you had a first child.
Yes.
You had a
Yes.
daughter,
Yes. Yes. A daughter. And then
born 1 year before leaving
that's right.
UCL. Because I've got a
Well,
feeling that you said they were marvellous
to-
because we had to take baby in and people helped us to
Yes,
kind
our
of-
colleagues were extremely supportive, most generous and yes.
Because I was
kind
interested
to our
in how
children.
one coped with having a
Yes,
family at the same time as being
yes.
so committed to laboratory work.
Well,
and you said
yes.
it was relatively easy because people were supportive.
Yes, yes. We didn't take the children in every day. Obviously, we had an au pair girl in the flat in
Yeah,
London
sure.
who looked after them most of the time. But when we did take them in, and when the first one was tiny, obviously we took her in.
But you had to have a son
And,
soon afterwards when
yes,
you got to
yes.
when you got to
No,
Camden.
people were very
Yeah. It's nice to have
supportive.
that part of the story
yes,
taken
yes.
in.
No, they really were.
And you got quite interested in motherhood, particularly not because I think you interviewed some mums around who'd also kind of had babies around the time of yours about their pregnancy experience.
With not so much the pregnancy experience, but it was the question of embryo transfer.
Right.
Because since we were doing embryo transfers in animals at that time, it was obvious that eventually this technique would become available for treatment for infertility in humans. I didn't know how long it would take. In fact, Louise Brown wasn't born until 1978, so that was 20 years later. But the technique was there, it was going to be extended to farm animals, and it was interesting to think of how the social and ethical implications would figure when it was applied clinically. And since one's friends were having their families at the same time, I remember asking one or two women, would they feel the baby was more their own if it was an egg from somebody else and they gestated the baby, or alternatively, whether it was their egg that they gave to what we would now call a surrogate mother,
Yeah.
and it was gestated by another woman. And I mean, a tiny sample, it can't have been more than 4 or 5, but most people said they'd feel it was more their own baby if they gestated it and gave birth to it. And I think I'd have felt that as well.
And that mirrors the kind of findings elsewhere in over more recent years. So there's a little bit of social research there going on in the background, apart from
I was always interested
fascinating.
in, well,
Yes.
you know, we were working for the Agricultural Research Council. I was always interested in possible applications of one's work. I think that was why I was working on mice and not on Drosophila or worms or anything else.
In this early, kind of moving with this early ball, if I can use a sports term, I mean, you were quite early in this field.
We were the
wave
first people in Britain to do any embryo transfer, and that meant, in fact, that it was very difficult to get the technique established because we had nobody to go to and say, show us what a mouse egg looks like, show us what an 8-cell embryo looks like, how do we get them out of the reproductive tract?
You had to make all your own mistakes.
That's right, that's right. But we struggled on, and while we were at the Royal Veterinary College, John Biggers was working there too.
Yes, next lab.
And he had a nice culture system going for chick bone. He was working on chick embryo bones, growth in culture. And so as we had these early mouse embryos, he cultured some of these embryos from 8-cell stage up to blastocyst. And then I put them back into the uterus of a foster mother, and they were born as normal, healthy mice, and we mated them and they were fertile. And that was actually the first time it had been shown that mammalian embryos could be cultured in vitro outside the body and they would develop normally thereafter. So that was an interesting experiment. And he
that must have been a spectacular time, and that must have caused a lot of publicity.
Yes, we had
around.
quite a lot of publicity about that.
There's one thing we've not looked at, and
Yeah.
I just think we might cover as we go. You were looking at a wave that hadn't beached, I mean, but the prospects of it all
Yes.
were quite clear to you, I think.
Yes.
That this would be a massive thing for medicine and have worldwide implications for fertility, for childbirth.
I hoped that it would be possible for the technique. I knew that Bob Edwards, who was, you know, a student at the same time that I was, he was also interested in early embryo development and embryo transfer. He was doing the same sort of thing.
Where was he based at that stage?
Edinburgh. And then he came down to Cambridge and started working on the human reproductive system. And you know, I had a great admiration for him, so my guess was that, you know, he'd pull it off and that one would in the end see Louise Brown being born, but of course I didn't know how long it would take.
Classical story, isn't it, though?
Yeah.
The momentum building up,
Yes.
all towards this incredible situation we have at present.
And all through the late 60s and 1970s, I'm going on a little bit now, there was a lot of discussion on social and ethical implications of the new reproductive technologies.
Yes.
It didn't come
And
as
you
a
must
total
have served on
surprise.
committees about that,
Indeed.
being central to that.
Yes, yes.
And we might put a major committee or two on the record, because there were critical committees. I'm thinking Warnock, is that wrong?
That was later. That was well
later,
after
yes,
Louise
but
Brown.
Thinking
yes.
before.
I remember there was a British Association committee. That must have been early 1970s.
Yes, 70s.
I
yes.
think it was Walter Bodmer who chaired it. Shirley Williams was a member. We met in the, what's that liberal club called in Pall Mall, round the corner from the Athenaeum.
Yes, it's famous.
London club.
Where a film that I remember started out, I think, from there,
right.
around the world in whatever days.
Well, Shirley and I had to be smuggled up through the tradesman's entrance and the back lift, because of course women weren't allowed.
Yeah. The Reform Club, is it?
Reform Club, that's the name of it, yes, that's right, yes, yes.
Just while we're
But that
talking
was
about
later.
Shirley Williams
Yes,
and politicians,
yes.
perhaps I could make a cul-de-sac kind of tour and just say that you were not immune from political interest. You
No,
got a
I was
strong
interested in
political
politics
sense.
and always very left-wing, because when I went to Oxford, it was that 1945 election,
Yeah.
And I followed that very closely. And I was delighted when Labour got into office.
Did you took part in campaigning?
I was more of an observer at that stage.
Right.
And then the big campaigns after that were against atom bomb tests throughout the 1950s. And I was going around lecturing on genetic effects of radiation, long-term effects of radiation.
I think whilst you were
Ban
in
the
Oxford,
bomb, all of that.
yeah, you'd set up a peace committee, helped to set up a peace
Oxford
committee.
District Peace Committee,
You ought to
yes,
have that.
yes.
Just looking at this
yes,
political animal that's
yes,
in there, Anne.
yes, yes.
Because it was strong. You did feel strongly about it.
Well, I still do.
Yeah,
Yes, yes,
but I'm talking about
yes,
then.
Yes,
I'm
sure.
just trying to get that time
Sure.
absolutely crystallised out.
Yes, yes, yes.
It
No,
was-
there was
it
a lot
was-
of
it
political
was
activity.
Very strong.
Yes.
How did that go down with the family, all this socialism? Because
I
don't
didn't
get-
see much of the family at that
Right.
time. The Oxford and District Peace Committee was quite active.
Dorothy Hodgkin was involved, I
Yes,
think.
yes, she was a member of it.
This was the
Yes.
first time you met Dorothy.
Yes, that's right.
It's
That's
incredible
right. Yes,
to
yes.
meet this
Yes.
soul who was also
Yes,
dedicated politically
yes,
to this
yes,
thing.
yes. In fact, that was those were the only years when I really saw anything of her,
Yeah.
because our paths
Golden
diverged
moment.
after that. Yes,
Yeah.
yes.
But this was the political soul. I just wanted
Yes,
to tease some
yes,
of that out.
yes, yes.
We'll get you back now to the research, but the responsibilities of this committee work, that must have been quite a happy part of being political and being involved, the wider field, the ethics,
Yes.
the implications
Yes,
down
yes,
the line.
Yes, yes, yes. It all interacted. But going back to the lumbar vertebrae, if I may,
Yeah.
we struggled on and collected our data. We devised a technique where we didn't actually have to kill the mice to count the number of lumbar vertebrae. We could X-ray them.
Right.
We used to stick them down on their backs with sellotape on the X-ray film and X-ray them in big batches. And we collected quite a lot of data, and it had to be analysed very carefully statistically. And Donald was extremely good at that, of course, because all his Bletchley Park experience was statistics oriented, because each litter had to be analysed separately. And there were complications because sometimes you got an asymmetrical mouse, which was where the sixth lumbar vertebra was sacralised on one side and not on the other.
Right.
But it was frustrating because the data really wasn't making a great deal of sense, and it wasn't telling us either yes, it's uterine or yes, it's cytoplasmic. And that's always a frustrating time. And then one day we were sitting in a pub chatting and realised that we hadn't allowed for the sex effect, because there's a difference between males and females in the degree of sacralisation as well as the degree of asymmetry. And when we looked at the data again and allowed for that, it made much better sense, and it turned out that it was highly significantly a uterine effect. So it was an effect exerted through the uterus of the mother and not the cytoplasm of the egg.
Did you go on at that stage to try to look at what that effect might be?
Well, by that time it was the end of the 1950s, the end of our grant period. We checked that it wasn't simply growth rate or anything like that. It wasn't related with size. I checked that it wasn't the body temperature of the two strains. There was no difference that was relevant there. And really, I think even if we hadn't moved away, there would have been no way in which at that time one could have solved it.
Technologically, the story wasn't there.
Because it wasn't really until the late 1980s that all the question about the Hox genes, homeobox genes, which lay down the pattern of segmentation of the vertebral column, emerged. Nowadays, one knows just which Hox gene it is. I think it's group 11 Hox genes that determine the sacralisation or otherwise of that sixth lumbar vertebra. And my guess is that today, if somebody went back and looked at that, they might find that perhaps there was a subtle difference in timing of development between the two strains, or it's been shown that that particular HOX gene, the expression is responsive to retinoic acid. So maybe there's a difference in retinoic acid or some related metabolite coming from the mother, which is different between the two strains. Something of that sort.
But as you say, the sync, the timing,
could
availability...
never have done that in the 1960s.
Colossal, you couldn't have possibly got near it.
No.
It's still not known.
That's right.
Yeah.
So I'm glad I didn't go banging my head against that brick wall.
It was back to your old style, though. Like, I'm not going to read all the Dickens, I'm going to do something else.
Yeah.
You put it aside.
Yes, I'm afraid I'm not a sticker in that sense.
The chemistry I put aside.
If I come up against a big problem, I prefer to go sideways. It has, yes. I've been very
come
fortunate.
back to two issues that we got into that would be nice to know about from that period. We talked about the superovulation and the super pregnancies.
Yes.
Can we just have outcomes on that, because that took part. He was going to go on. It was going to span into your next kind of existence in another research centre, but some results had come out in that period of the 50s.
Right. Well, we were comparing the number of eggs shed by young females in response to hormones, older females, and the extent to which those eggs would successfully implant in the uterus and whether you could get pregnancies resulting. And then, yes, when I moved to Edinburgh, I continued that work, and I looked at the degree to which eggs were shed from right ovary versus left ovary, the asymmetry business, and the relationship with placental weight. So there was a lot of later work on that, and of course my job in Edinburgh was again on the staff of the Agricultural Research Council, and they were interested in ovulation.
Yes, from a moment, but in the results, I think you found that exactly as one might expect, the casualty rate really mirrored very much the overloading of the uterine environment. Yes. In fact, the greater the number of embryos implanted, the smaller the number of babies born alive. One got a U-shaped curve. Right.
Up to a point it increased, but then after that it decreased again. So from the point of view of agricultural productivity, one didn't want to push up ovulation rate too high.
Yeah. We can get to 1959 when you go north.
When I moved to Edinburgh,
Scotland,
yes.
Let's just tidy up the edges of the London existence before we travel. You're a mum, 3 children by now.
Well, the last was born in Edinburgh. Yes.
In Edinburgh.
Yes.
But this is part of the plot by now. Marriage is coming to an end.
Yes. And scientifically, we were going our different ways.
Right. So there's a major bifurcation.
Yes.
Of a team that was.
Yes.
Donald went off to work in the University in Edinburgh.
That's right, the Department of Surgical Science for Professor Woodruff, Michael Woodruff, on tissue transplantation.
I should say that I think the thing that surprised and excited me in looking at that family relationship you've had is that it still continued.
Yes,
and it
indeed.
still was immensely fruitful and supportive and meaningful to both of you over the years.
Yes, no, we went on being good friends.
Yeah.
Yes.
Classically important relationship that goes on and goes on and goes on.
Yes.
Whereas, well, let's get you to the Institute of Animal Genetics. Waddington's brainchild, is that right?
Professor Waddington, yes.
He's an amazing character.
He was my boss, indeed.
I think underestimated, am I right?
I agree. I think he isn't appreciated scientifically nearly as much as he should have been.
Yeah. But you'll make that right, because you have a...
Well, today I've been interested that quite a lot of his lines of work are now becoming fashionable again, and people are referring to his early work in the literature.
You mentioned Waddington earlier when we talked this morning and said that he was one of the names in the literature who was an exciting writer at the time.
Right, on evolution.
Yeah.
Yes. Well, of course, that was well before I knew him.
So you were already in touch with his work and thinking to some extent.
Yes.
You went there. Was that an exciting institute to join? I mean, what was the stage in 59?
Enormously exciting.
Yeah.
No, it was the most wonderful scientific atmosphere I've come across anywhere, partly because of the intellectual excitement. What Waddington did was to get grants from all over the place. I mean, probably more than 100 grants came into that institute. And he put them all in a big pot and stirred them up and used them for whatever he thought was most in need. And one of the things he did was to subsidize the canteen there. And at that time, I think it was the only canteen on the campus, and people used to come from all over, zoology, molecular biology, and there were these wonderful lunchtime conversations. Yes,
So
tremendously intellectually stimulating. I mean, quite apart from the people in the institute itself.
And was Waddington
quantitative
present?
genetics.
Was he around? I mean,
He was abroad a lot.
right.
So
I just got a feeling he was always into something.
Yes. No,
So he wasn't a natural part of that stimulating conversational environment.
Not so much. I
Yes.
mean, when he was there, he was very stimulating, but he wasn't there much.
But he was the top man, a powerful administrator.
And he organized the institute in such a way that everything was laid on. There was a graphics department, a draftsman who used to draw diagrams and beautiful drawings of mouse embryos. There was an excellent photography department, statistics, excellent statistics, expertise, very good library, of course. Histology was done for one.
This was ahead of its time.
There was even a film unit. So if one wanted time-lapse cinematography of one's embryos developing, one went along to Eric Lucy and got the film done.
It happened.
Yep, yeah. Very good animal facilities, mouse, rabbit. Yeah, it really was an amazing institute. Early computers, the lot. And of course, when I left to set up my own unit, I realized for the first time just how fortunate I'd been, and that all these facilities didn't grow on trees. If one wanted them, one had to set them up oneself.
I'm just looking to this Conrad Waddington, was it?
He was always called Wad,
Yeah.
but I think his initial CH actually stood for Conrad Hal.
He took all these grants.
Yep.
Like a supremo.
Mm-hmm.
I mean, I think he'd been a head of bomber commando, done something in the war and been quite powerful in the area.
It was
Took all
what's
this
called
money.
operations research, and I never knew quite what it was, but it was obviously very important.
Yeah, he took all this money
Mm-hmm.
and dished it out as he felt fit.
That was the impression that I got.
And other people got that impression because he got into bad light
I'm afraid he
subsequently.
did, yes, I'm afraid
Was
the
this with the research
accountants.
councils? Oh, right.
Yes, I think they
Because
finally...
it didn't end well. I've got a feeling it didn't end well.
No, he got into some trouble, I think. I don't know the details. Yes.
But that's sad because it deserved
Yes.
a better epitaph.
Yes.
Let's take-
But as I say,
Yeah.
his reputation is now
Yes.
very high, very high.
Yeah. Let us take in the research that you went with then. You were still committed to the maternal influence field
Well, yes.
and
yes,
the super ovulation.
the reproductive biology,
Yeah.
the super ovulation, the maternal influence. I did some work on immunocontraception, immunizing
Right.
mice against sperm, immunizing female mice against sperm, which induces infertility. And that again is a technique that much later was taken up for possible clinical use. I mean, it's still under investigation as a possible new approach to contraception.
Do you think that has real mileage? You feel that that's got a large
Yes.
future?
Whether it'll be immunization against sperm antigen rather than one of the other antigens that are being tried, HCG, that's human chorionic gonadotrophin, may be the most optimistic one, or zona pellucida. There are a number of different approaches, but I think that the immunological approach is a promising one. Yes.
So this fertility area became the field you
Yes.
increasingly were known for.
And with
Yeah.
Waddington's help, I and Alan Beattie, who was working in the Institute, got a big grant from the Ford Foundation for reproductive work, possibly leading to new forms of contraception. And
It didn't go into the pot. You managed to keep.
yes. And of course that enabled me to take on a postdoc or two and a graduate student or two. And I did a lot of work on implantation, because the implantation of the embryo in the uterus is a very critical point of reproduction. Certainly in the human, it's the time at which a lot of pregnancies fail, and therefore it's a promising target for contraception, as well as an important aspect of infertility.
Yeah.
And I did a lot of work on that in mice. I was unable to identify the signal that the mouse embryo gives to the uterus, which induces the first implantation reaction, which is crucial to the success of implantation in the mouse. And actually that still hasn't been identified. Yeah. And partly because I was up against a brick wall there, and partly because I realized that the mouse model of implantation was not a good one for the human. It's quite a different type of implantation, pattern of implantation, that I moved on and gave that up and went more back to the...
But it's a classical challenge, isn't it? What is
yes.
the message, the signal that heralds this incredible response on the part of the uterus
That's
to accommodate
right.
a blastula?
I knew what the systemic message was that would sensitize the whole uterus. Right. That was estrogen, yes. which was derived from the ovaries as a result of the mating signal. But what the actual local signal for each blastocyst in the uterus was is still not known. And yet it's very, very definite, starts at a particular time. I could identify 3, 4, 5, 6 stages immediately following on.
Really?
Yes. But
we've got you think we've got a classical receptor ligand kind of sequence?
I think we have, but of course in those days one didn't know about receptors, one didn't know about ligands, there was no molecular biology.
Yeah, hasn't it come a long way? We thought it
certainly has.
Like the names of receptors could be counted on one finger.
Yes, yes, yes. No, it was a very different scene. I tried to get into DNA because in the early 1960s I went to a congress of zoology in Washington, D.C. and...
In 1961, I think.
61, that's right. And heard Roy Britton talking about the Britton and Davidson DNA hybridization work that they were doing.
Yes.
And I thought that was absolutely fascinating, because it seemed to me that if one could hybridize DNA from different sources, it would be like an antibody-antigen reaction, and one could do the same sort of subtractive hybridization that one can do with antibody-antigen, and maybe pick up, differences in the DNA between species that were too distant to be able to cross them sexually. And I worked for quite a while with Peter Walker in Edinburgh. I was able to enthuse him when I got back to Edinburgh, and he was very, very helpful and spent a lot of time with me trying to make this work. And we did get some results.
Yeah, he was in the university working on the molecular
He was in the zoology
side,
department, yes. That's right. But unfortunately, the technology in those days was only capable of identifying hybridization based on highly repeated sequences. And of course, those aren't the sort of differences between species that I was interested in. So although it was interesting and useful experience, it didn't lead to very much.
But philosophically, you got deeply tied into the possibilities of that.
Yes, indeed.
I mean,
that's
that was
right.
an exciting period.
Yes,
and
it was.
Could have gone miles if there'd been opportunity.
Yes.
It eventually came good because Ed Sutherland...
That's right. Ed Sutherland was one of Peter Walker's postdocs at that time, and it was shortly after that he invented southern blotting, which nobody now hardly even remembers. The southern of southern blotting is Ed Sutherland's surname. Yeah.
So those were fascinating early 60s.
Yes.
And the DNA work just got going 2 or 3 years. I think you were regularly going over to the university. It was quite a busy time.
Yes, all through the mid-60s.
Yeah.
Yes.
What actually followed in the later 60s? What came to take its place?
Well, I was doing embryo culture back in the genetics department, the same sort of culture that John Biggers had started off doing at the Veterinary College.
So
he'd moved to America and was carrying on with that work in America. And I was very interested in the new technique of making chimeras, where you aggregate embryos from two different strains together to make a compound embryo, twice the normal size, but it regulates in size and is born the normal size.
And of course, if you regulates gastrulation somehow, is that right?
Yes, that's right. What seems to happen is that one whole cell division is inhibited, switched off. Yeah. So it, from being twice the normal size, gets back to normal size
back
again.
into line.
But it's clever that it knows how big it ought to be, isn't it?
Yeah.
Yes. Yes.
But that must have been absolutely, I mean, that must have taken over a bit.
Yes, well, it meant getting the culture technique set up first, because unless you have a good culture technique, you can't possibly make chimeras. And once the culture technique was working and the embryos were growing in culture, then we were able to start making aggregation chimeras. And I had this strain of mice which had 8 or 9 different Mendelian recessive mutations in it. And so chimeras between those embryos and normal wild-type embryos were extremely interesting from all sorts of points of view.
Take me into that story a little bit, Anne. Give me a little bit more on that, Anne.
Well, let me see. Obviously, the first thing one looked at was the pigmentation, the coat colour, because that's the most striking thing you see in mice and the easiest to look at.
Can you get zones? You can get zone effects.
Yes,
really
they're patchy.
Pronounced.
They're patchy. That's right. And
that goes with hair as well. I think you said you were counting hair and granules of pigment.
Yes, one can look at individual hairs, and one could see the chimerism even within the individual hair. A single hair could have some of the characteristics of the mutant strain and some of the wild type strain. And some of the genes act through the pigment granules themselves, but some act through the environment of the hair follicle. So again, you get an interaction between the gene expression and the environment in which it's operating. The agouti gene, for example, is expressed through the hair follicle, but the albino gene is expressed through the actual pigment granules, the pigment cells.
You were really into a field that was very much where you'd look to be. I mean, this was cells surrounded by environments that were quite curiously variable. A cell normally knows what's around it, and there's very great consistency.
That's right.
All of a sudden you were stacking around cells, new environments.
That's right, that's right.
That's very much your developmental area.
In a normal animal, every cell is surrounded by other cells of the same genetic constitution. But here one could look at cells that were surrounded by other cells of a different genetic constitution, and that gave some very fascinating results. I looked at the coat colour, at the individual hairs. With Grunewald in London, we looked at teeth, and we looked at skeletal characteristics. And then I looked at the reproductive tract, and that was the thing that led me on to subsequent work. Because, of course, half the aggregation chimeras you make are going to be, on average, between a female embryo and a male embryo. And most of those develop as males, which is interesting in itself. A few develop as intersexes, and a few develop as females. And following up that story, and also looking at how the germ cells, deciding whether they're going to turn into sperm or eggs, were affected by that environmental interaction, that proved a fascinating line of work throughout really the 70s and 80s.
Right.
Yes.
So that went right on.
Yes.
Yes. But classically important to fertility research.
Yeah. Yes, yes. And very interesting to me.
Grunewald and that work on teeth, that was quite remarkable.
Grunewald was an amazing man. He was a meticulous worker who knew every corner of every bone in the mouse body and every cusp on every molar and tooth. And I would never have had the patience or the knowledge to have worked that out. but with his help we got some quite interesting results.
Fascinating strains you got of mice with curious teeth.
Yes, they were the tabby mice.
Tabby mice.
Yes, they have funny teeth. Yeah.
Thanks for putting that on the record for me.
Yeah.
And we're getting you through the 70s. We've already indicated that things were to go on into the 80s.
Right.
This was the great pattern of the 70s though, that we've got into. The chimera story was the great part of the second half of the 60s going into the 70s.
That's right. Yes. Yes.
How did that I think you went about 1974 to a new
Well, it was all going very well in Edinburgh, and because it was going well, Alan Beattie and I thought that we could ask the Ford Foundation, rather impertinently, for a large sum of money to build a centre of reproductive biology in Edinburgh. And so we wrote up a big grant application, and they gave us $250,000. And at the same time, the Medical Research Council were wanting to set up a unit of reproductive biology. And those two schemes came together. Edinburgh, through a bid that was put together by David Baird and Roger Short, they won the bid for the MRC unit of reproductive biology. So the four of us got together and decided that the centre should be built in Edinburgh and should house the unit. And that sort of plotting was the early 70s. But of course, in the way things are, it took a long time to be actually planned and built. And meantime, I had been attracted to London, partly because my children were getting to university age and the centre of gravity of the family was moving south. And I'd been in Edinburgh for 15 years, which is a long time to be in one place. So the Medical Research Council set up a new unit, and I became director of it. It was called the Mammalian Development Unit. Those were the early days of computer databases. I wasn't allowed more than 2 names in the title of the unit, otherwise it would have been called something rather different, I think. But so it was called Mammalian Development, and we had all sorts of funny envelopes addressed to mammary development, and I don't know what else. But anyway,
where was that set up, Anne?
It was set up at University College in the same mouse space that Grüneberg had recently vacated, because his unit had come to an end.
So you're eventually coming back to UCL?
That's right. And I'd, of course, visited Grüneberg in connection with our joint work on the chimeras, and so I knew his mouse space was good mouse space, and I went and looked at it and measured it. And so we moved in there. Unfortunately, and this was a shame really, unfortunately the Lister Institute packed up at just about the time that we were moving in, and the Medical Research Council rescued the very important blood group unit of Race and Sanger, and there was nowhere else to house it except in our space, so we gave up 1/3 of our space to the blood group unit.
So you sacrificed right away?
That's right. So the unit was never actually on the scale that I would have liked, and that made problems with accommodation all the way through. Yeah.
Because you got it all set in your mind by then.
That's right, yes, yes.
What was the time you came down? Was it 74 you came?
74,
south?
yes, that's right.
Back to London?
Yes, back to London.
And at that particular point, getting you back to London, I'm going to leave it for today.
Right.
And we'll take up the story of the later years
and
all their fascination.
Okay.
Next time we meet.
Fine. Well, thank you very much.
Thank you very much.